Welcome back to my course, that industrial biotechnology. Now, in the last lecture, I tried to cover spirulina. One thing I forgot to mention that we 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 have one book that is algal biorefinery and integrated approach. So I suggest that if you want the information about the algal production processes, you can get the detailed information in this book. Now, let me come back to the this lecture that we know that different enzymes is very much used in the day-to-day life, and one of the very important enzyme is alpha amylase, which has a lot of applications now in the different industry, and also it is used as a for in the medicine, not only used for in some food making industry, but also used in the medicine for medicinal purpose. Now, this alpha amylase, we have this among the industrial important enzymes like protease, that amylase are considered the most prominent enzymes, since they are widely utilized in the brewing, detergent and food industry. Because we know that in the detergent industry, I told you that where sometimes we have some blood clot in our cloth, and this is blood is kind of protein molecule. So if we use some kind of protease enzyme, that will clean this clot, and sometimes we have some kind of strain due to some fat type of material. We use some kind of lipase that also do the it can remove the strain, and sometimes the starchy material also has some kind of color the contribution to our cloth, that also removed by amylase. So this is exactly what is mentioned here, that used in the detergent food industry is largely used. I think we are going to talk about the high fructose corn syrup production, that there I shall tell you more. And brewing industry, already we have seen how brewing industry, because the first steps in the brewing industry is the malting process, and through the malting process actually we develop a lot of hydrolytic enzymes like alpha amylase and protease. These enzymes are produced during the germination process. After the World War II, the enzyme application increases due to the advances in industrial biotechnology and biochemical engineering, and amylase is an enzyme that catalyzes and hydrolyzes the starch into sugar, and amylase is present in the saliva of human, because we know our saliva contains the kind of amylase enzyme. Some other mammals where it begins the chemical process of digestion. So we have three different type of amylases. We have alpha amylase, beta amylase and gamma amylase. We will be concentrating on mostly on alpha amylase today. And if you look at alpha amylase is cleaved alpha 1,4 linkage. We know the starchy molecule is a polymer of glucose, and they binds with each other by alpha 1,4 linkages, and this is alpha amylase cleave the alpha 1,4 glycosidic bond present in the inner part of the amylose amylopectin chain of the starch. Starch has two type of chain. One is called amylose. Amylose means the starch molecules, they form, this is the chain, this is the chain molecules, and in this is amylose, and this is glucose molecule. This is the polymer of glucose. Now in case of amylopectin, we have the branching. We can have some branching here. So we have, this is called amylopectin. So this bond is alpha 1,4 linkage, and this bond is alpha 1,6 linkage. So they have different type of linkage that we have, and alpha amylase calcium is very much required for the activity of the enzyme. We observed that thermostability of the alpha amylase enzyme increases to a great extent in presence of calcium ion. So alpha amylase can be used for two purposes: or saccharifying alpha amylase. One is liquefying alpha amylase. The saccharifying alpha amylase, they hydrolyze 50 to 60% of glycosidic linkage of starch. Liquefying alpha amylase clip about 30 to 40% of glycosidic linkage of the starch. This is the two type of alpha amylase, and this is they called endo amylase. Endo amylase means that this can randomly attack the starch molecule at any position. You see that the any position here, here, here. So in process it produces oligosaccharide. Oligosaccharide means it is maybe dimer, trimer, tetramer, different this hexose molecules. They in combination they produce the different type of oligosaccharide that you get that as a product due to the action of this alpha amylase. And then we have other type of amylase, the beta amylase. The beta amylase are they walk alpha 1,4 the glycosidic bond, but only on the non-reducing end of the polysaccharides, and making it slow and clip two glucose off at a time, resulting a single maltose molecule. So I can show you this. It is beta amylase, largely available in bacteria and fungi, and pH is about 4 to 5. Now if you look at here, that this is the non-reducing end, and here the this beta amylase can also, so the two glucose unit can comes out. Here also two glucose units can. We know that this two glucose unit, when they bind with alpha 1,4 linkage, we call is amylose. So this is amylose is the main product. That then gamma amylase is the clip the alpha 1,6 glycoside. I have already pointed out that this bond is alpha 1,6 glycosidic linkage, and this is alpha 1,4 glycosidic linkage. This is the difference that we have in reclaiming the, in addition to 1,4 linkage at the non-reducing end of amylose and amylopene resulting glucose. So gamma amylase is more efficient in acidic environment, has an optimum pH in 3. So history of amylose production is like this. History amylose begin in the year 1811, when the first starch degrading enzyme was discovered by Kirchhoff in wheat. Then alpha amylase was named as Kuhn this in 1925, because the hydrolysis products are alpha having the alpha configuration. Then amylase was first enzyme to be discovered and isolated by Payen in 1833. Then interestingly, first enzyme produced industrially was an amylase from fungal source in 1894, which was used pharmaceutical aid for the treatment of digestive disorder. We know during I told you that the amylase has lot of medicinal applications. When we use we have a digestive problem, that doctor usually prescribes some kind of tonic which contain amylase and proteases that help in our digestion system. The Bodin and Effond in this 1917 were the first to use the Bacillus subtilis and Bacillus mesentericus for the production of alpha amylase on commercial scales using the large fermenter by submerged fermentation process. The industrial production of alpha amylase, the plant source, they play a crucial role in the plant growth regulation. Beta amylase appears prior to alpha amylase to initiate the germination process. Plant sources are not being considered with enough significance, as the source of this enzyme become low productivity and difficult to cultivate. The animal source, we have like Tyline, that is an alpha amylase, that is one of the most important enzyme in the saliva. That I told you, in the saliva we have this amylase enzyme. When we take the food, they mix with our food, and starchy material will degrade. Then the alpha amylase based is produced from microbial source, both from bacteria and fungi. This for industrial production, it is cost effective, is a consistency, less production time, is in the process modification and optimization. The bacterial amylases, we have several. We have Bacillus subtilis, we have Bacillus thermophilus, we have Bacillus licheniformis, Bacillus amyloleucophacins that are known to produce a good amount of alpha amylase. Thermostable enzymes isolate from thermophilic bacteria, having the commercial application due to stability. Now, bacterial enzymes, they are exploited commercially due to their rapid growth, leading to shorter fermentation cycle capable of secreting the protein into the extracellular media and safe handling. Now, here I want to point out that all the hydrolytic enzymes produced by the microorganism, they are usually the extracellular in nature. So, that doesn't presence inside the cells. So, it presents in the fermentation media. So, you can separate the cell mass, and in the fermentation broth you will get that, in the filtrate you will get that enzymes. So, you can purify it. So, this is the enzymes produce considerable quantity, mostly from Aspergillus species and the Penicillium species. Then like Aspergillus oryza, Aspergillus niger, and then this pH tolerance is less than 3. That is there. Filamentous fungi is suitable for microbial in solid state fermentation process, especially because their morphology allow them to colonize and penetrate the solid surface. Now, this gives you a table that indicates that what is the mode of fermentation for different type of microorganisms, and what is the optimum pH required for this fermentation process, what is the temperature required, and what is the molecular weight of enzyme that we produce by this, and what are the different inhibitor is present. This bacterial alpha amylase that produced from this has different characteristics. Now, factor influencing the production of alpha amylase is the selection of microorganism that plays very important role. I told you the industrial strain should higher productivity. The temperature of the media plays very important role. pH of the media plays very important role. Composition of the media, carbon nitrogen source, incubation period, inoculum age, and agitation and aeration. These are the couple of things that plays very important role for alpha amylase production. Now, if you look at the selection for the industrial enzyme, criteria is that specificity and reaction rate and pH, temperature optima, and stability. The stability of the enzyme plays very important role as per industry concern, and stability of the enzyme is usually determined from the half-life of the enzyme. So, usually we prescribe the half-life of the enzyme should be as high as possible. Effect of inhibitor, how inhibitor, whether it is easily that affects that activity of the enzyme, that is also very important. Affinity to substrate. Now, if you look at the process flow diagram for this enzyme production is here. You have different microbial strain. We have submerged. I already explained you the submerged fermentation and solid state fermentation process. In the submerged fermentation, microorganism grow throughout the liquid, and solid state fermentation, that microorganism also grow throughout the solid media, that this is usually in the liquid media. Then this is the cell separation that take place, cell free, this is the biomass. It can be used as a compost. I told you that this alpha amylase is the extracellular product. It comes out of the cells, so it present in the supernatant. You can make it concentrate, and then you stabilize by adding certain ingredients in it, substrate analog or some other material, and then this product has usually in the market, we do the market. Similarly, we do it from this solid state fermentation process. Now, industrial production of alpha amylase by bacteria, it is produced either solid state or by submerged fermentation process, as I mentioned before. And due to rapid growth of bacillus species, it is more predominant in the production of amylase in the industrial purpose. Then most commonly used commercial enzyme is Bacillus subtilis and Bacillus licheniformis and Bacillus amyloleucophacins. liquefacence. This is the mostly used by the industry. These strains are improved by mutation to yield the enzyme about 250 times greater than the wild strain. Different techniques such as the cell immobilization in the matrix such as allisinate, polyacrylamide, agar and kappa carrageenan are being used for continuous and rapid batch fermentation of amylase. So, this is for the degradation of starch. We use that. Media use mostly the complex that wheat bran and rice bran, this is gram hugs, the mustard oils, oilseed cake, which known as the agricultural waste or agricultural waste is used. Most of the strain undergo the catabolite repression with the glucose. The glucose is the undesirable product in this because glucose gives some inhibition effect. The yeast extract has been proven to be the most preferred organic nitrogen source for alpha amylase production as reported by many studies. Then yeast extracts not only serve for the nitrogen source but also rich source of vitamin and other growth factors. The thiamine, cysteine and pyridoxine are the amino acid it found mostly suitable for alpha amylase production. The media, we have phosphate concentration of the media has a regulatory effect on enzyme production by beta amyloliquefacence. A high phosphate concentration promotes the maltose uptake and the microbial growth, while a high maltose uptake rate suppresses the enzyme biosynthesis due to the catabolite repression effect. Thus, the phosphate concentration should be optimized in the cultivation broth. The production by submerged state method required the medium optimization including 5% starch, ammonium nitrate, sodium citrate, magnesium sulfate, H2O support, and calcium hydroxide 2H2O, and then yeast extract, etc. Then inoculum preparation. Inoculum is prepared in the sec flux, then inoculum is grown in the small fermenter, then finally in the production fermenter. The optimum pH of the production is the 6 to 8, and temperature maximum enzyme production occurs at relatively lower temperature 27 to 30 degrees centigrade, but it depends on the bacterial strain used. The thermophilic bacteria such as the Thermonospora species produces maximum enzyme when the temperature is 53 degrees centigrade. Aeration is done in the range of 0.8 to 1 ppm. The duration of this fermentation is about 48 hours because this is the bacterial fermentation. This is usually takes less time. If you go for the fungal fermentation process, it will take little longer time. The enzyme fermentation rate is slow during the exponential growth phase of the growth phase, and just before the rate growth decreases, the spore formation begins, amylase production increases. So, here you can visualize from here that enzyme, this is the enzyme production, this is the cell mass concentration, that is cell mass that is growing like this. So, after sometimes when that cell mass grows is maximum and sporulation starts, then enzyme production increases. The production of fungal amylase, similarly particularly Aspergillus oryza is used. Mostly it is produced by using the solid state fermentation process. The wheat bran is moisture and steamed 1 to 2 hours to extract the soluble starch. The substrate contains 8% starch and the sodium nitrate and magnesium sulfate, KCl and ferrous sulfate malt extracts are added to optimize the media. Inoculum fungal spores are the fungal, I told you that fungal spores is used in the solid media to use as the inoculum. That production of fungal enzyme temperature is 28 to 30 degrees centigrade. The duration time is 3 to 5 days. I told you in the case of bacterial fermentation it is 2 days. So it takes quite long time, 3 to 5 days. Fungal molds grow specially designed tray equipped with perforated covers and high rate of aeration provided they encourage the growth, because this is the aerobic fermentation process. Now product recovery is like this: the fermentation broth is separated into liquid and solid part by means of filtration and centrifugation. This is the fermentation broth. We give some kind of, we add some kind of 2% diatomaceous earth. This is kind of filter aid, then vacuum filtration, then we put ethanol, and precipitation of enzyme take place with the filtration. Supernatant we discard the precipitation, we vacuum dry for 8 hours, grinding in powder, packaging moisture proof container, we get the amylase, and this is used for the waste treatment processes. So this is for fungal amylase, filtration alone is sufficient to separate the solid from the liquid, while the bacterial amylase filtration and centrifugation are carried out. This is you can see the product that has been marketed in the different forms. The marketed of amylase, that either in the liquid concentrate or powder. Liquid concentrate is preferred in the industry due to the convenience of a handling, because when you mix with this, liquid can mix thoroughly, but when you use solid, then you have to make in suspension. So that is very important. It also form an important part of digestive enzymes. So you can see this is a powder form, and this is a liquid form. This is again powder form that has been marketed. So the application of alpha amylase is tremendous. The sugar and glucose industry is largely used. Then we have used the alcohol industry. I told you that IFB agro industry, which is located close to Kharagpur, that they produce alcohol from broken rice, which is used as a starch source. And starch is a grain, potato, etc. is used as raw material to help the manufacture of ethanol, is the first converted in the fermentable sugar. In the textile industry, to hydrolyze and soluble starch for increasing the stiffness of the finished product. So this is required, and fabric size with starch. Amylase is used for desizing purpose. So application of the bread and chapati industry is largely used to make it soft, because you know that more than 70% bread USA, Russia and European country contains alpha amylase, so that we can easily digest it. And alpha amylase degrade the damaged starch of the wheat flour into the small dextrins, which allows the yeast to work continuously during the dough fermentation and proving and early stage of baking. The chocolate industry, the amylase is treated with coca slurry to produce the chocolate syrup. Treatment of starch processing wastewater, this analysis of medicinal and clinical chemistry, it is largely used. The increasing of stability of alpha amylase plays very important role. The production of stable amylase is a high temperature. I told you calcium plays very important important effect on that acidic pH and calcium independence are successfully achieved by using the following approach, that production of the extremophiles organism, because if you use the extremophile which can grow at a very high temperature, and it is expected the enzyme produced by this organism, they are quite stable at high temperature by using the recombinant enzyme, protein engineering, amino acid mutagenesis and chemical stabilization methods. I told you by using some kind of substrate analog, or it is possible to increase the stability of the enzyme, and metal ion stabilization methods and immobilization method largely used for the stability of the enzyme. So in this lecture I try to discuss its special topic, that is the alpha amylase production, and which is largely used both in the food industry as well as in the pharmaceutical industry. Thank you very much.